Today is the first of 3 talks by candidates for our position in evolution and cancer. The idea is to find people taking advantage of what turns out to be very rapid progress, trying to understand how malignancies evolve and possibly finding ways to prevent that evolution by decreasing the resistance to chemotherapy agents, plus many other possible angles on evolution and cancer. We're very glad to have with us today Robert Getzenberg, who's Director of Strategic Clinical Affairs from GTx Incorporated in Memphis. Prior to that, he had a very long career at Hopkins, where he's Director of Research for prostate cancer, among other problems. He has previously done a postdoc at Yale and done training there. He's been looking for markers, especially for cancers, possibly in blood and other places, to try to find easier ways of making a better diagnosis. Today's talk is titled Novel Approaches to Lethal Resistance of Advanced Prostate Cancer, Stress in the Cancer Microenvironment. Welcome.
Thank you very much. Well, thank you. It's a pleasure to be here today. What I'm going to try to do is accomplish a number of things and take you through a sort of at least some of my thinking and ideas and some data to support it, hopefully, about how I feel that evolution is critical to our understanding of cancer and certainly towards developing new ways to identify and personalize cancer therapy, as well as new therapeutic approaches. And the only thing I didn't check is how to advance the slides. So first of all, I want to go through it and talk a little bit about why are we having such a problem in cancer? Why is cancer such a tough one to lick? And we'll talk a little bit about successes in cancer. Oh, great. Thank you so much. No? That's an NEC one. I'll keep hitting this. It's fine. We've had a lot of successes in cancer, but we've had a lot of where we haven't had progress. And you'd think with our understanding today, we'd make a lot more impact. Why have we not? Talk a little bit about variation. That's obviously one of the critical aspects of cancer. And I'll tie in a little bit of heat here as a microenvironmental factor and how I think it can be used as a cancer therapeutic approach. So this goes back, and the slide's a little bit dated now, but it's actually not that different than where we are today, except for the number of drugs has increased significantly. But we really have not made that much impact on cancer mortality over the years. It's gone down a little bit, and the big successes we've been talking about over the last couple years have really been small inflection points in these numbers, in my opinion, but we really haven't had the type of impact that we think we should have. Richard Nixon started the war on cancer in 1975, the RAS oncogene 1981. We've done a lot of molecular understanding of cancer to date, but yet where is this translating into impact as far as patients' lives? And this, again, a couple years outdated now, but over this was a 25-year window of time. There were 131 new drugs developed, tested, FDA approved, marketed, all these molecular targets identified, but yet the impact as far as cancer mortality has not yet been seen to the extent we have. And again, this number of new drugs has increased significantly over the last couple years, and we're seeing some little blips in there, but why can't we lick this cancer problem? What are really the issues there? Well, one of the things is cancer is not one disease, right? It's many diseases, and some of them are actually much more lethal than others. So if you just do something very simple, and this was my just taking basically the number of deaths per year versus number of new diagnoses, it's a figure I made out of those numbers. You can actually see the spectrum everywhere from pancreas cancer, where the lethality is actually quite high as 85% or greater, all the way through to some of the common ones, colons here, breast, prostate, one I work on a lot, right there, and testicular cancer. And we've often used that as a model, and I'll talk more about that upcoming in the talk, which is, why is there such a difference? Why is pancreas cancer so lethal and testicular cancer on this end so curable? And what can we learn from those that we can use to impact the other ones in the middle here? So these are the kind of thinking I think that's important to really understanding cancer and looking at cancer in a new way that can really be therapeutically useful. So resistance is really the biggest problem in cancer. Cancer cells develop a resistance usually pretty rapidly to any kind of therapeutic agent that you're going to give them. And we'll talk about why that is in a second. But it's really the big problem in cancer. You have chemotherapeutic agents. Almost all of them are cytotoxic, right? So they kill cells. So in theory, if you gave enough of them, you should be able to get rid of all the cancer cells. And it's the balance between killing the cancer cells and not killing the normal cells that really is the issue in cancer therapy. And the other problem is that normal cells don't develop this resistance. They're much more sensitive to many of these agents, And therefore, the side effects or the issues that go along with using cancer agents is actually quite significant. So the therapeutic index is very important in understanding how much agent we can give, whereas we can get rid of the cancer cells without disturbing the normal environment. And so I am not an evolutionary person, so that'll be my caveat to start with. I am someone that's come late to thinking of evolution, and I've tried to come up with my understanding of evolution and applied it to cancer. So I'm not a classical evolutionist, if that's a proper word. But obviously evolution is critical to everything we do in biology, and hopefully I'll convince you in cancer. In technology, we select for the best things in technology as well. But let's focus today on cancer and trying to understand that. So again, an oversimplicity, but yet I think hopefully able to demonstrate where I think we are in cancer today. Obviously, this was from a paper published in 2007. All the dogs out there in the world, all the different breeds came from one single wolf, and we used artificial selection to then select out different characteristics, whether that was size or ability to hunt or swim or whatever you wanted to do, to end up with all the different things. In this case, what they showed is that a change on chromosome 15 was selected for that was encoding insulin growth factor, and that's what gave the difference there. But this is the same type of principle in cancer therapy. We end up with a cancer, a tumor cell, and yet it ends up with many different types of phenotypes. So how do we go after that? How do we attack that? How can we stop this resistance from happening where we don't end up with all these different species? At the same time, the original one, right, was quite stable. The Siberian wolf has been stable for 1 million years. But we've selected out all these different subpopulations by using selective breeding and looking for characteristics that we found are attractive. So what I want to do next is talk a little bit about how we might identify and characterize some of the castrate-resistant cells prior to hormonal manipulation. What does this mean? In prostate cancer, prostate is a hormonally sensitive organ, and the hormones, the androgens, are often withdrawn in prostate cancer as a form of treatment. Resistance happens just like it does with cytotoxics in almost all patients. So how do we prevent that, and how do we identify it up front? And are there markers that might allow us to help stratify that? So again, one of the problems with cancer is heterogeneity. We talk about cancer as a breast cancer, a prostate, a lung, or a colon, but they're actually quite complex. In fact, this is a slide from a single patient, and you can see here they have different types of prostate cancers within there. Here's one type here, it's a little out of focus, but here's another type over there. And pathologist Don Gleason actually used a drawing to show the different forms, and the more de-differentiated, the further you are down on the scale, down on 5, the more aggressive you are thought to be. So it's not just one prostate cancer, this is actually multiple things. So if we do a tumor genome sequence, what are we sequencing? Are we sequencing this one? Are we sequencing that one? Are we sequencing a mixture of the both of them? Or none of the above? So this heterogeneity has really been a big problem in helping to signature and fingerprint cancers, and that's really held us back from really understanding what they are and developing better tools to identify them. So we did this experiment, which will be obvious to those that are not in the medical or cancer area, but actually I think was very liberating, at least they were illuminating to us, which is we tried to understand, for example, in this population of prostate cancer cells, do androgen dependent and independent cells exist prior to any selection, or is our selection procedure selecting for them? So are they already naturally there, or do we select for them? And so we took actually a population of cells and did this experiment. The idea was to go back to that slide I just showed you where we had different prostate cancers there and ask, did they all exist prior to that, or did somehow the therapeutic procedure begin to select for some of these? So we did a simple experiment. We took a prostate cancer cell line called LNCAP. This is a human cell line that came from a lymph node. That's where the LN came from a prostate cancer patient. And basically what we did is just plate these out. We cloned them out till there were single cells per well. We let the wells grow up, and then we began to pick the clones. This is before we did any challenge to them. So this is just taking the individual cells, growing them up in wells to see what happened. In this case, we just took a relatively small number just to get a sampling of what was going on. And what we found out was something very interesting. So here's the P as the parental line, and these are androgen sensitive, so if we put them in charcoal strip media, they stop growing basically. So that's the normal pattern, the parental line. So we had clones that looked very similar to that, like clone 5, clone 3, looked very much similar to the parental line. But we also had clones like clone 1 here, where it continued to grow even in the absence of androgens. So this clone existed prior to our selection pressure and already was there with resistance before we did anything to it. And so you could see this clone 1 continued to express a marker of androgens, PSA. It had the androgen receptor, and it was able to continue to grow even in an environment where there were no androgens existing, whereas the parental line did not grow. And so what happens when we compare these? Again, this is PSA expression. Both of them make PSA. This is the clone 1, the one that's androgen independent or less resistant to the absence of androgens. And if we get rid of them, it's still sensitive, so the androgen receptor is still working there, but something in the pathway is different between these. Because if the androgen receptor wasn't working, it wouldn't change at all, depending on whether we put them in normal media or charcoal strip media. The other thing we did is used a drug called bicalutamide. This is an inhibitor of the androgen receptor, and again, it's still responsive to that. So the receptor is still working, so it hasn't just turned it off or acting promiscuously, but it's actually responding the way it should respond. But these cells continue to grow even in the absence of androgens. Interestingly, these cells, this clone 1, are larger, so they're actually physically bigger cells than both the parental line as well as some of the other clones that we looked at. Clone 5, again, is an example of one that behaves like the parental line. So they're physically bigger, so there's a different biology going on there. And when we look at their invasive properties, what we find is that they're less invasive. Although they grow in the absence of androgens, they're less invasive than the clone 5, the one that looks like the parental line, both in vitro as well as in vivo, where the clone 1 is down here. It doesn't grow as well in vivo, and the clone 5, the one that's more like the parental line, continues to grow in animals. So although they grow differently under hormonally stressed conditions, this does not necessarily translate as far as their invasive properties in vitro or in vivo in animal model systems. When we looked at the genomic patterns that were going on related to their chromosomes, we used the SNP array, and what we found is that they're not the same. There are genomic differences between clone 1 and clone 5. Again, clone 1 is the resistant line, clone 5 is the one that is sensitive. And what we can see here are there's certainly some differences in chromosomes gains and losses that are going on between these two. What did we look at when we look at a microarray analysis using, again, the same different clones? We found genes that were both found at higher levels in clone 1, but also some that are at lower levels in clone 1, and tried to focus on a couple of those that we thought might be instructive as trying to understand what is found in the parental line that might be missing in clone 1 or vice versa. That might be telling us something important about the biology here. So we focused on these two that were most highly overexpressed, and I'll tell you a little bit about those in the upcoming slides. So first one is called Sprouty. Okay, this was originally identified as a Drosophila gene. That's an antagonist of FGR signaling, and it's found during trachea development. It's downregulated in a subset of prostate cancer. So here's some data here from other studies in prostate cancer where people have shown that it's actually downregulated in prostate cancer. And its expression levels have been associated with biochemical recurrence of prostate cancer as well. And it's been a target, as I'll show you, we have some data for as well, for microRNA 21. So that microRNA is able to mediate its expression. The other one's JAGID1. So JAGID1 binds Notch receptor and plays a role in epithelial mesenchyme transitions in cancer. And its expression is also associated with prostate cancer, metastasis, and aggressiveness. Again, it's been shown to downregulate, its downregulation can inhibit prostate cancer growth. So a number of studies have been done in that. Well, you can see it's more highly expressed in metastatic ones than it is in benign tissue or localized prostate cancer. So what happened in these clones related to these genes? Can we confirm the earlier results? So again, we went back to look at the parental line and then the two example clones we had, clone 1, which was the resistant one, and clone 5. And interestingly, in my mind, clone 1 and the parental line looked much more similar than they did in the previous analysis. Again, this may have to do with just the populations and how you isolate them, but there are still significant differences between them, whereas clone 5 now shows to be significantly different. And you can see here that Sprouty was much more highly expressed, as we anticipated with the microarray analysis, in clone 5 than it is in clone 1. both at the RNA as well as at the protein level shown here, and supported by the immunofluorescence studies. One of my colleagues, Sean Leupold at Johns Hopkins, had been working on miR21 and found that both of these genes have miR21 sequences, so they were able to show that they could be regulated with those. And what we basically then did is to do a series of experiments to see how does this really impact the expression of these. So what we basically did in this study was to then express miR21 in both of the clones to see what happens. So basically, if we express miR21, no, we looked at the miR21 expression, in both of these and showed that miR21 expression is higher in clone 1 than it is in clone 5. So that may be obvious because based on the expression patterns that we saw. And then what we did is to look at miR21 in these LNCAP cells, parental lines. When you then turned on these with androgens using an artificial androgen, R1881. So taking them all the way from no R1881, that's the 0, all the way up to 10 nanomolar, which are pretty high levels. And you can see here in LNCAP cells, the miR21 goes up, and then the Sprouty levels at least go down. The JAG1 levels go down to a much lesser extent. Again, that's RNA levels. And when we look at the protein levels, what we can see here is that miR21 can also be associated with expression of a downregulation of the Sprouty as well as JAG1 as well. So again, the Sprouty is a little more dramatic than the JAG1 is, but both of them are downregulated with miR21. So to try to put this together in a model, what we basically showed here is that the androgen receptor expression seems to be altered between clone 1 and clone 5, and we think that miR21 at least may be one of the drivers of this, and this may be causing the Sprouty and the JAG1 expression to be lower in clone 1 than it is in clone 5. The question is, how does this then translate to the activity we see at the end of the day, which is the AR activity, as well as both on the proliferation effects, which were different in that clone 1 grows in the absence of androgens, whereas clone 5 does not, as well as the invasive properties where it's vice versa. So we still have a lot to figure out here, but at least it gave us some suggestion that these proteins may be involved in the differential of these two different clones to be able to grow within these populations. Finally, I like to always look at and see what does this mean to what's happening really in patients with prostate cancer. So what we did is to do an analysis here to see what the expression was in several different types of prostate cancer patients. Here we looked at Sprouty expression, again looking at mRNA, comparing people that had PSA failure or not. So after surgery, some people have PSA levels increasing, which amounts to the fact that they have metastatic or advanced disease. In this case, Sprouty levels were lower in this population of patients than they were in the patients without that. Gleason score, again, is this pathology score. In this case, JAG1 was higher, more highly expressed in those cases than it was in the others. And then PSA levels, and in fact, PSA levels are lower in higher Gleason scores. This may be counterintuitive to people when they think about PSA levels going up in men with advanced prostate cancer, but the reality is the more dedifferentiated the cells are, the lower on a per cell basis PSA levels are. So again, some of the results we're finding do indeed translate to things that we're observing in patient settings as well. So just to summarize this first section, I think we've been able to show that, first of all, and most importantly in my mind, there were LNCAP clones. And we always look at these cell lines as sort of one model of uniform cells, but they're not. They're heterogeneous just like the tumors are themselves. And they have different levels of androgen independence that's already existing prior to any selection pressure that's put on them or not. The LNCAP clone that we looked at with higher AR activity and higher androgen independence had lower invasiveness and in vivo tumor growth. Not necessarily intuitive, but that's what we observed in the study. A DNA copy number variants exist between these, showing that there's some differences. And there's obviously two of the genes that were differentially regulated are Sprouty and JAG1 that were differentially expressed between the two different clones. And both of these have been shown to be regulated by miR21, an androgen-regulated microRNA, and that Sprouty1 can negatively regulate AR activation. I didn't show you some of the data on EGF-induced AR-independent cell proliferation because of time, but we've done some additional work looking at EGF in this pathway as well. So hopefully that gives you some sense for where we're going with some of those studies. In the next section, I'm going to talk a little bit about the effects of resistance on response to microenvironmental stresses. And so the goal here is to look at microecologies as physically induced stressors. And I'll explain a little bit more about what I mean in a second. Then we also want to examine the responses of normal and cancer cells to these same stresses and determine if there's a difference in responsiveness to cells that develop resistance. And that's one of the key features here. So getting back to what I started with, one of the things I think it's critical to understand in cancer is where has our success stories been? And this is an example. We published a paper called The Lance Armstrong Effect that got published in JAMA maybe 6-7 years ago, where we tried to look at testicular cancer and why it might be curable versus other cancer types that are not curable. And one of the ideas we had, and this may sound silly, but there's actually some data to support it, is that the testes are normally cooler than the rest of the body, as they're several degrees cooler. In fact, if they're not, and you have undescended testes, they don't descend properly, they actually increase their risk for cancer. But because they're cooler, the temperature differential between where they normally exist and then they metastasize inside the body might be significant enough that they now become more susceptible to cancer therapy. And so the question is, how do we begin to apply this to other solid tumor therapies, and how do we really go after this question to see if it's right? This became one of the big questions proposed by Harold Varmus to look at in cancer, which is why are some cancers curable and others are not? But certainly a question that still exists today. So one idea is that temperature plays a big role in this difference. And I think this is a good quote that I saw a couple years ago that I really like, that it's important for us to think about cancer in a different way. It's not just about what's going on with the cells. And cancer was no more of a disease of cells than it is a traffic jam is a disease of cars. And a lifetime study of the combustion engine would not help anyone to understand our traffic problems today. And the cause of congestions can be many. A traffic jam's due to failure of the normal relationship between the cars and their environment, and can occur whether they themselves are running normally or not. So it has to do with both the cells, but also the environment that these cells are in. And so I think this was an important and insightful quote that was generated from 1962. And so one of the things I want to talk to you a little bit about is heat and heat's importance in life. I think heat is, and again, I'm not going to go into all the aspects of this, but heat self-organizes DNA. It controls differentiation of stem cells. It's obviously a very important and powerful microenvironmental tool, and it's also can be a sex determinant, obviously, in reptiles and other animals. And just to give you a really sort of overly simplistic example of the importance of heat, if you take a fertilized egg and you leave it on the countertop here at 70 degrees at a room temperature for 21 days, nothing happens, right? Eventually the egg will rot. But if we do one thing, which is to add heat to it, what you can begin to get in 21 days at 100 degrees is a chicken. So that's the importance of heat. It separates everything from a rotten egg to a chicken. And temperature effects can enhance a lot of different things. Immunotherapy, you get fevers, and that's important in fighting off different diseases. It's been shown to make radiation therapies work better, as well as chemotherapy as well. And so in order to modify temperature, it's important to understand how cancer cells respond to temperature. And then how can we look at some of the molecular changes that are going on as a response to heat that might be important. Obviously, heat shock proteins are those that are stimulated by heat. Cold shock proteins are those on the other end of the spectrum that can be turned off as temperatures are warmed. But both of those are sort of the regulators of what's going on within the cell. We use a lot of different cell lines to study these, everything from normal prostate models to those that are much more aggressive. Thankfully, there's now becoming much better characterized cell lines. Everything from the PREC cell, which represents essentially a normal prostate cell, to an OPCT cell line, which is really the localized prostate cancer cell line, to some of the ones like I just talked about, the LNCAP cell, and then more invasive ones like PC3 and DU145. And I'll show you some examples where some of those cell lines have been selected for long periods of time, in this case in the presence of taxol, to be resistant to chemotherapy. And we can now use those models to understand the difference between resistant and sensitive lines. So what happens when you heat cells? And so what we did is to heat 3 different cell lines, PC3 and LNCAP, 2 different prostate lines, and also a testicular cancer cell line, and just took them from 37 to 41 degrees centigrade, and just to see what the differences is. We wanted to look for big expression differences and ones that were consistent, and we ended up with a series of 38 genes at the intersection there. So again, what we found from this is obviously heat shock proteins, right? And so those are the most obvious ones. There's actually a couple drugs now focused on heat shock proteins in clinical trials. We also found a really interesting family of proteins that I knew almost nothing about when we started these experiments, which were these RNA binding proteins that were really downregulated when you now heated up these cells. So what are these? And a couple of them were examples were RBM3 and CIRBP, and I'll show you some data from this RBM3 protein, which is really intriguing. So these are the molecular regulators of what's going on in the cell level at the temperature. And I think understanding these may give us more clues about how cells respond to stress and how this may give us new therapeutic or biomarker targets. So what is this RBM3? Well, this was considered to be a new class of proto-oncogenes in a paper published in 2010. It's been shown to regulate microRNA biogenesis at the dicer step. It enhances global protein synthesis. It's been shown to regulate gene expression in a circadian fashion by controlling alternate polyadenylation, and regulates phenotypic switching via alternative splicing. So it does lots of different things, at least reported in the literature. None of this is our work. So what we did then is to begin to say, well, what happens in prostate cancer models, and what's the expression of this RBM3? So we looked at normal organ-confined prostate cancer and then metastasis, and found it overexpressed in organ-confined prostate cancer here, but actually much lower in metastasis. And we have another protein I'll show you like that as well. There's a couple of these that are coming up that are atypical in that they seem to be turned off in metastases, but are elevated in expression in localized cancer. And I think these are very intriguing, both as far as what we know about cancer and the difference between a cancer that can grow in the local environment but yet cannot metastasize, and some steps that may need to be turned off in order to become a metastatic one. We also showed that the CD133 expression in CD133 positive cells, it's almost not expressed at all versus CD133 negative cells. And so I think we're beginning, and also when we looked at different tissues, for example, this is normal prostate, fetal prostate. It's much lower in fetal prostate. Normal testis, fetal testis. So the fetal tissues express much lower levels of this protein than the adult tissues did as well. What happens when we begin to look at whether this is expressed in either more aggressive model system, which is that grown in agar conditions, versus a 2D regular cell culture system? So this protein in two different cell lines, both the PC3 and DU145, is expressed at much higher levels in the normal cell culture conditions of two-dimensional culture versus the 3D soft agar condition. So again, another model showing that this may be down-regulated when you put this into an environment where it's more aggressive or needs to act more aggressively. Another feature that was reflective of the CD133 expression is the stem-like properties of this. So what we began to do then is to look at these colonies that grew. So you can see here, I don't even have to show it quantitatively, but when we grew these colonies, this is the RBM3 overexpressors. These are the normal ones. You can see here they barely grow at all, and obviously very few colonies. And we also tried another model, which is a 2D model where you make these prostospheres, which are supposed to represent sort of an in vivo kind of prostate that's happening, but on a cell media. And you basically are letting them grow into three-dimensional clones. You can see here we really got none of those. So these cells that overexpress RBM3 are not able to carry out those functions. So we seem to have taken away their ability to grow in that fashion. What happens if we inject these into animal models? So again, took RBM3 overexpressors, put them into a mouse model, and again, you don't need to even look at the quantitative data. In one of the lines that we used, we never got any tumors. In the other, we got a couple small tumors in a couple of the animals, but the rest of them did not express any. So really showing that this is a pretty good restrictor of tumor growth and aggressive growth by overexpressing RBM3. This RBM3 overexpression upregulates a number of different things, particularly differentiation-related genes, as represented by this section over here. So these are upregulated. compared to the parental line, the control line. And this series of genes, which are really related to stemness or self-renewal, are all downregulated. So again, what we're trying to show is that RBM3 is inducing this more differentiated, less aggressive type state. And if we turned it off, you basically would become less differentiated and more aggressive. It also has been shown to alter CD44 expression, and we did a whole series of experiments related to CD44 expression. This is important in reflecting the type of prostate cancer cell that it's being derived when we modulate RBM3 by overexpressing it. And just to show you here that the expression pattern of CD44 is very different in the RBM3 overexpressors than it is in the control lines. So just some conclusions from this section. We know that temperature-induced cell changes are very important. Again, obviously the heat shock work has been very important for a long time. I'll show you some other work to look at nuclear matrix changes as well, but didn't go over that because of time. But RBM3 really it represents a cold shock protein that really may modulate this process. I've not seen as dramatic of a data even with some of the heat shock proteins in this regard. Alteration of these RBM3 levels influences tumor growth and may synergize the effects of chemotherapeutic agents. We have some data to support that. Again, I didn't show it to you for time as well. And RBM3 levels decreased with the stem-like properties of the cancer cell. So the next question is, what fitness price do cancer cells play as they develop resistance, and how can we take advantage of that? So when we utilize prostate cancer cell lines, what kind of sensitivities develop at all? And what are the molecular changes that accompany the development of resistance, and how can we take advantage of that? So again, going back to that list of cell models I used, we used two models. One was a DU145 and one's the PC3. Both of these are metastatic lines, and both of them were grown for more than a year in taxol conditions, so with Paclitaxel chemotherapy. And so obviously the blue line represents the Paclitaxel resistant line. The red one shows the sensitive, where you kill off a large percentage of the cells. The PC3 is resistant, but not as resistant as DU145. If you give enough, you can get a lot of them to die off, but certainly much more resistant than the parental line is, which is the red line below. So these are two models that were available to us. So we went back to that heat idea and said, well, what happens if we use heat? Does it synergize at all? So we first asked a simpler question. If we took the normal prostate cells, PREC cells, and the primary prostate cancers, do we see a difference? And you can see here, if just heating them at 42 degrees for a period of hours, you begin to see that the prostate cancer cells do not do as well as the normal cells do, though that difference is not really that big, but it is certainly real. When we look at the androgen responsive and androgen insensitive, again, the insensitive is a more aggressive resistant line. You can see here that the androgen insensitive line, in this case, was more sensitive to temperature than the androgen responsive line. Again, more similar to the normal conditions I just showed you. So in both of these examples, the more normal-like you are, the more you are less sensitive to temperature, and the more aggressive or more cancer-like you are, the more sensitive you are to temperatures. Let's see what happens in these resistant lines. In the resistant lines, this is the PC3 line here. You can see that the Paclitaxel resistance is very sensitive to temperature, so it actually kills off quite a good percentage of the cells. And the DU145 line is also sensitive, though again, it's one of those things that both of them, if we let them go long enough, we begin to see the lines come together. But certainly, there is a big difference in sensitivity to temperature between these resistant lines. So we did an experiment, which is to say, if we took these and combined them together, how does that work? And so what we basically did is to take these different lines here, and you can see here everything from the organ confined, the LNCAP. This is a androgen resistant line of LNCAP. The PC3 cells, and they're taxol resistant. The DU145s, and they're taxol resistant, to see what we can do as far as. So you can see here that with combination, in this case, even if we gave the drug, it really doesn't affect the cells at all. But if we heat them up at the same time, we can really get rid of most of them. So the concept here is, can we use a combination of a drug that we know these cells are going to be resistant to, but at the same time, are they paying a price? And this price is a sensitivity to a microenvironmental condition, in this case temperature, that they're much more sensitive to. And by combining a classic cytotoxic agent with a microenvironmental change, can we now get a synergistic effect. So we've done a lot of our work looking at nuclear structure and the nuclear matrix. This is the organizing structure where all the DNA is organized within the cell, and really, in my mind, one of the centralizing themes to cancer. One of the questions is, what does the nuclear structure look like when you look at the resistant and sensitive lines? And we've looked enough at these patterns that they don't just look like stars in the sky to us. We can actually see some patterns reflective here. And what we saw was a series of proteins missing in the resistant ones that were found in the sensitive. So when we looked at the resistant, they were missing. And we began to say, well, what might those proteins be? And as we sequenced them, what we found is that they were actually represented cytokeratin 18. So cytokeratin 18 expression, and this was just the nuclear matrix proteins I just showed you, but this is with a 1D gel. The cytokeratin 18 is much more lowly, or expressed at a much lower level in the resistant cells than it is in the parental cell line. And when we go to look at cell lysates, it's even more dramatic. You really can't find it there. So why would these resistant cells be turning off cytokeratin 18, and what's going on in the biology here? So John Kim, who was an undergrad who worked in our lab for a couple years when I was at Hopkins, who's now a biomedical engineering Ph.D. student at Berkeley with a joint program with UCSF, did, I think, a couple really neat experiments, which was to look at not just cytokeratin 18, but the other cytokeratins to see what was happening. And what he saw in this case was in the more resistant DU145s, these cytokeratins were really turned off. 8, 18, 19 are really turned off. In PC3, this is the less resistant one, they're downregulated. You know, certainly cytokeratin 18's downregulated, 8's downregulated, 19's about the same, but certainly they're affected, but much more so in the DU145 cell that I just showed you. And what the idea is, what else might be going along with this? So one of the things that cytokeratin changes have been associated with is a transition from epithelial to mesenchymal state. And so other changes that have been associated with this represent some of the proteins outlined here, like vimentin, E-cadherins now. And what you can see here is there's definitely some differences in expression here. This ZEB1, which is a marker of EMT, is turned on. Vimentin is expressed now. This is a mesenchymal marker. SNAIL. Catenin, beta-catenin, and epithelial markers turned off or lowered in expression. So you see now a bunch of changes reflecting of this as epithelial to mesenchyme transition that's going on within this pathway. So far, what it seems to us is as the cells developed resistance and are more sensitive to heat, they've become more mesenchymal-like in nature. And this may reflect their more aggressive nature, their ability to become more metastatic, etc. So what does it look like? Is that true? Are they more metastatic when we look at these lines? So this is just a scratch assay where basically you take the cells, you just take a pipette tip and you scratch through it and you see how fast they grow back. You can see here, this is an invasion. So if they just keep growing, they'll pile up, but if they invade, they grow in together. In 2 days, the taxol-resistant ones have grown in together, whereas the parental line basically stays apart. So they're much more invasive from that. When we look at invasion in soft agar, what you can see here is obviously, again, a lot of colonies here. The parental line did not form any colonies. And invasive assays, where you basically look at the ability to invade through a membrane, you can see here there's much more invasive activity in this taxol-resistant cell line. Finally, what happens in an animal model? So if you put these into a mouse, you can see here that the taxol-resistant one grew to much larger tumors, whereas the parental line did not form tumors. These are sub-Q implantation of the cells, or grew tumors that took much longer to develop and are much more smaller. So these are much more aggressive tumors. So the evolution of resistance to Paclitaxel increases sensitivity to microenvironmental stresses. I showed you heat, but a similar thing's true for caloric restriction, pH changes, etc. We've published on this and we have additional data on this as well. I think a very interesting area and one that reflects perhaps some different approaches that can be taken to cancer therapy. And I think developing and understanding the mechanisms that underlie these is also critical to our really optimizing and using these different treatment approaches and really understanding what resistance is and how we can attack it. So hopefully in this part what I've shown you is that evolution of resistance to therapy is one of the biggest problems in cancer treatment, and that development of resistance to chemotherapy makes them more susceptible to some of these microenvironmental stresses. And for the rest of the talk, what I want to do is focus on a series of proteins called cancer testis antigens that I think are very important connections between the testis and cancers. I think they're involved both in the cancer cell and its mechanisms as well as its interaction with its microenvironment, and we think they can be utilized to characterize different phenotypes of prostate cancer. So what are cancer testis antigens? These are a group of proteins with diverse functions that are predominantly expressed in the adult testis, but have been aberrantly expressed in a bunch of cancers now. They've been used as immunotherapy targets for a number of cancer types, not all successfully, so a couple of melanoma studies, I think one of which recently failed in the last month or so. But obviously these are a unique set of antigenic proteins that seem to be relatively cancer specific. Very interestingly, there's a set of these that are found on the X chromosome. Not too many things of dramatic interest are found on the X chromosome, with the exception of a couple things we're interested in prostate cancer. But these CTX proteins are found predominantly by their name, found all on the X chromosomes, and they're actually quite interesting. They are typically, they constitute multi-gene families, and they don't seem to have orthologs in other mammals, so they seem to be relatively new in the evolutionary scale. So here's where they seem to have appeared. Again, here's Homo sapiens. Here they're relatively new as far as evolution in principle, and I think we could talk a little bit about why that may be important in development of cancer as well. They have a lot of similarities between embryonic and development as well as spermatogenesis and tumorigenesis, and we've tried to focus on some that might have characteristics in all of these, but they obviously have a lot of things in common that I think really relate to these cancer testis antigens, which are in the middle there. So what are some of the functions? They actually have a lot of diverse functions, everything from centromere, chromatin modulators, transcriptional regulators. You can see the list here. So they carry out lots of different things, particularly related to DNA organization and as well as cell signaling. We put together a nice circus plot looking at all the known 228 known cancer testis antigens, and they're showing basically their chromosomal localizations. You can see here, these are the CTX, the X chromosome overrepresentative of the cancer testis antigens. And again, this is obviously of direct importance to our work in prostate cancer. A majority of these are intrinsically disordered proteins. And what are those? So intrinsically disordered proteins, or IDPs, are proteins that don't have a stable tertiary structure. So I did not learn about these in graduate school, but are a very interesting series of proteins. They have no defined structure like the one on the left does. They're intrinsically disordered, as their name implies, and they really get their shape based on what they bind to. So they're basically like master keys. They can almost open any door, and they get the shape of whatever they're fitting into. And this shape is very important, obviously, to their function and importance in obviously both normal function and diseases. So here's an example here where if you take the folding of one of these, this is a transcription factor, CREB, and you bind it into its protein, and now develops a structure based on the protein it's binding to. And so really that's what intrinsically disordered proteins is. They have no structure until they come and bind into their target site, and that develops their structure. IDPs are prone to a lot of things, but one of the things that's very interesting, particularly with what we're talking about here, is they can promote what appears to be promiscuous interactions. So they can bind to lots of things. So here, for example, is P53. It can complex with many different things, and in doing so develop lots of different structures based on the kinds of proteins that it binds to. They're also more likely to occupy hub positions. These are central positions in signaling, just like hubs are airports. This is US Airways hub. These are hub positions in the protein networks, and these are very important in determining signaling pathways. And we've been thinking a lot about how these hub networks may be disrupted in cancer as well. Cancer is also is a phase transition, which all the things we're talking about have often been shown to be reversible. This epithelial to mesenchymal transition can be reversible. The stem cell and non-stem cell-like nature of cancer cells can be reversible. Possibly the sensitivity to drug resistance can be reversible. So we believe these phase transitions may be reflected with some of these intrinsically disordered proteins and how they're binding to different proteins and carrying out functions. So we've developed a series of these, discovered that are associated with prostate cancer. For example, we have several here that this is the relative expression that are found to be expressed in benign tissue. So for example, page 4 is one we've done a lot of work on, really interesting. It's highly expressed in benign, in primary prostate cancer, but not expressed at all in metastasis. So really intriguing. Why does it have to be turned off in metastasis, and what's so important about that? Again, another one, GTSP1, that's expressed benign in primary. And then we have a series that are found in metastatic ones. So between this, can we develop a series of markers that might be useful in identifying and separating the different cancer types? So Dr. Sarashi, whose picture I just showed you, worked on developing a G signature and then using a separate set of samples, looked at some of these same markers, and basically built a custom microarray. And based on the training set I showed you, then tested it in another set using a TACMAN assay, showing that these are indeed found in metastatic tissue, whereas that page 4 I showed you is only in primary and not in metastatic. So we're beginning to put together a series of markers that seem to be predictive of different types of prostate cancers. And so then the question is, how do we take this to the next level and validate these results? So we then looked at a larger set of samples that we were able to obtain from another institution. And again, you can see here, this is the geo data. This is data that we've been able to generate with qPCR. This is looking at MAGE-2. This one's turned on in metastatic prostate cancer. Here's PAGE, again, with again supporting the previous data that this is not expressed at all in metastatic tissue. And so again, we can begin to put this pattern together and by weighting them, begin to develop a recurrence score that we can use to predict the risk of recurrence from this. So then just to show you how it came out, we were able to obtain 72 samples from patients with localized prostate cancer. 29 had recurrence of their disease, 43 did not. These came from Dr. June Liu at Johns Hopkins. And these are some of the different cancer testis antigens I talked about. Again, here's page 4. The levels are lower with recurrence. This is not the same as metastasis, so it's a different measurement. This is just whether the disease recurred, only looking at the primary tumor. But we definitely see some differences between these cancer testis antigens and whether the patients recurred with their disease or not. And in fact, we can develop Kaplan-Meier curves to show that we can differentiate these based on their cancer testis antigens as well. So what we're in the process of doing is building a multiplex qPCR assay, perhaps using nanowire technology where we can automate this approach much more rapidly and also evaluate formalin-fixed samples. The data I showed you prior was on fresh samples or frozen samples. We want to be able to do this on formalin-fixed samples like you'd get from a prostate biopsy. And then using that biopsy information, be able to show whether we can use our CTA gene signature and then develop a recurrence score to see whether we can predict who recurs with their disease or not. Again, a very important piece of information as patients consider whether they need to have surgery or not. Just to show you a couple slides on some of the biology here. This is work done by Yu Zhang, who was a postdoc in my lab. Again, this page 4 is a really interesting molecule. We talked a little bit about its expression. It's also expressed in the fetal prostate in addition to the local prostate I told you about. We've shown that it's expressed in BPH samples, which is on the top right there. It's not only in BPH, but highly symptomatic BPH. So this is disease where the patients have a lot of symptoms, need to undergo surgery, and actually one of the few markers of a more aggressive type of BPH that's ever been identified. Again, its dynamic expression is really interesting in the fact that it's not expressed in metastatic disease, but is expressed in localized prostate cancer, and that we can separate PSA-free survival, which is again is this recurrence based on the expression of this marker. Also, very interestingly, by turning off page 4, you can do a variety of things. First of all, you can show that the inhibition of page 4 in 3 different cell lines lowered the ability of the cells to grow and also made them more susceptible, in this case, to chemotherapy. In this case, it was adriamycin that he used. So it makes them much more susceptible to chemotherapy. So lowering expression seems to certainly have an impact on these cell lines. Also, administering using siRNA in SCID mice and LNCAP xenografts actually shows that you can get much lower or slower tumors being formed. Though they do grow, but they're much slower than they are before. And this is only using siRNA at the initial 3 time points shown here, and the rest of the time there's no administration. So by lowering this, by showing that you can block page 4 expression, what you can show is that you can significantly lower the tumor growth in animal models as well. It also protects, if we overexpress it, it protects them from stress-induced cell death. Again, adriamycin shown here. Basically, the page 4 overexpressing ones are resistant to adriamycin administration. This is, again, an intrinsically disordered protein. One of the things that we've been showing and working with Prakash Kulkarni, one of my colleagues, is that this interacts very well with June, and so it seems to be very important in June's transcriptional activity. So here's June alone. You can see here June in combination with page 4 gives you much more expression. Again, this idea that page 4 can come in and act as an activator of this by serving as an intrinsically disordered protein, and this may be the pathway that it's activating through here. So hopefully what I've showed you today is that we need novel approaches to cancer treatment. I think the Achilles heel of cancer may be its sensitivity to stress in the environment. Clearly this is evolutionary principles at work as far as developing resistance, and I think we need to understand both the mechanisms of resistance as well as how we can attack these and use them as markers of different types of disease as well. And I think cancer testis antigens are very important markers, both evolutionarily and biologically as far as the roles they play as intrinsically disordered proteins and in cancer. So I thank you for your attention. I thank my colleagues and funding that we've had to do this work. Hopefully you didn't go through too much. I was trying to give you a sampling for some of the areas that we've been interested in and some of the data that we've been able to produce in those areas. So thank you. And I'm supposed to click on something to get people. I don't know if anyone's there or not, so it doesn't.
Thanks very much.
Sure.
We can take a few questions about the talk first, and then anyone who asks, we can, and we can run a discussion.
Yes.
Do we really need it? Okay.
Otherwise, it will make no sense.
I see, okay.
Otherwise, they can't hear
How do
you.
you deal with the problem of genetic heterogeneity when you're doing your qPCR work, which seem very effective at detecting biomarkers?
Right. So in some ways, you can't avoid the heterogeneity, right? Because it's going to be there. Even if you take a metastatic lesion that looks homogeneous in nature, it's metastasized to the lung and it's a single nodule, you find heterogeneity even existing within there. So in this case, we chose to not ignore it, but to say that in order to get a marker that's going to work reproducibly and validated, you need to be able to have it work in conditions where you have the heterogeneity. So I think the discovery approach you can do where you try to make it as homogeneous as possible to find the most important markers, but then if they're going to work, you can't create that environment. It doesn't exist in the real world. So it has to be able to detect that population even in a heterogeneous environment. So in some ways, we've accepted that where we have to detect it in is heterogeneous, but we need to start with a homogeneous marker to begin with. And that's where I think the discovery process needs to focus in on a pure population asking a specific question, but then to test it needs to be heterogeneous because that's the real world. I hope, did that answer your question?
Yes.
So I'd like to ask more about page 4. So
Okay.
I may have missed part of it at the
Yeah,
end.
and I went
but-
through a lot fast at the end there, sorry about that.
Right, but so that was one that was low in metastatic.
Yep.
But then if you inhibit it, then you got increased growth.
Right.
Is in the model.
Yep.
So
So growth is not the same in metastasis.
Right.
So there's two different properties, right? So I think we showed you even from that clone 1 and clone 5 example in the beginning, those androgen sensitive and sensitive, that one grows better, right? But the other may metastasize, have more aggressive properties. And so I think that's what we're seeing here, that these are, for example, it's expressed in fetal where I think there's obviously growth going on, right? You're developing new cells, they're dividing, there's replication, but then they're not spreading all over the place. So to me, the difference is a more aggressive phenotype that's being developed. And that's what I think page 4 needs to be turned off in order to get. It can grow, but in order to get the more aggressive phenotype, you need to turn it off in order to do that. Does that make sense?
So then in that case, that would be one that's associated with and maybe stays in the capsule.
Correct. So some people have described it metastasis suppressors, right? So this could be, we have not done the work to definitively show this as a metastasis suppressor, but that type of concept where there's growth suppressors, metastasis suppressors, and they may not be the same thing. So there's obviously a lot of properties that need to go into developing a metastatic lesion to disconnect from your environment, be able to spread, reestablish, all those things. So it seems to me like that needs to be turned off, at least from this data, in the metastatic state. It doesn't mean it's not growing, it's not expressed in the growing ones. In fact, the BPHs where we find it more highly expressed grow very large and have a lot of proliferation. So there's clearly, in my mind, a disconnect between proliferation and metastatic ability. This, to me, is much more associated with an aggressive metastatic ability, the downregulation of it, than it is a growth. Does that make sense?
Yeah. So the other question I had was about the intrinsically disordered proteins.
Yes.
So can an individual intrinsically disordered protein bind to multiple? I mean, does it
Absolutely.
have a true partner, or does it bind to
Well,
multiple?
so people are still studying them, and there's a lot of work being done in this field. It's a growing area, but it appears that they can bind, they have many partners, but there seems to be some selectivity based on amino acid sequence. So they don't just bind to anything. So they're not out there just floating around binding to anything. So there is some specificity to it, but they have the ability to bind to often more than one partner. That's that hub position model I was trying to show you, which is the fact they can turn on and off many signals, but again, they're not turning on and off every signal. So there is a selectivity based on sequence there, but their polarity of their sequence is mainly what's contributing to the fact they don't really have any set tertiary structure, and that gives them this ability to, once they bind into a site, to sort of then conform to that site, but they still have a selectivity component to that.
So do IDPs have special motifs, amino acid motifs?
Yeah, they do, and
I
so
don't
you
know.
can actually search for them, and there's some algorithms now out there that I can lead you to you can search your favorite protein and see how intrinsically disordered it is, and you can get it on a scale from 0 to 100%. Basically, there's numbers you can attribute to it based on almost solely on sequence of amino acids, and mainly polarity is the key feature there. Yes, in the back.
So one of the most common sites of metastasis is the lung. The temperature in the lung is actually quite lower than temperature in internal organs, for example. So how do you reconcile that metastatic disease with this idea that the higher temperature is actually going to cause tumor regression?
Yeah, it's a good question. So I don't actually know that the temperature, is that a fact that the temperature in the lung is lower than the rest of the body?
Yeah.
Okay. Well, so that would be harder for me to reconcile, the fact that why you would be able to cure them. I would think that the cells that are within the lung, though, are still at 37 degrees. So even though the air that's getting in there is, the cancer cells are still 37 degrees, right? Or are they lower? I don't know. I don't know this.
Your argument is that the higher temperature is going to enhance their clearance, right?
Right. So
It's not just being at 37, it's being at 42 or something.
correct. So, but they're not, you're saying the temperature is lower in the lung than it is.
Well, it certainly is not going to be 42. It's going to be more like 37, so you're not going to see that enhanced effect.
No. So none of the body, right? So I guess maybe I didn't explain it clearly enough. So the idea with testicular cancer, for example, is the fact that the testicular cancer comes from an environment where it's several degrees cooler than 37. So it's 34 degrees normally, the testes. So now it's in the body at 37. That temperature differential is what's made it susceptible.
You see that same temperature differential in the lung
Going from where?
compared to internal organs, right? If you compare the temperature of the tissues in the lung, even though they're vascularized, the continual flow of air is actually cooling them quite
On the
a bit.
alveoli. But is the tumor are the metastases themselves at a lower temperature?
You can make the same argument, though, that they're cooler.
Right, they're
than-
cooler, but that does not necessarily make them more sensitive. So cooling doesn't necessarily make them more sensitive. It's really the raising the temperature that does. Though, because we've done some experiments in cooling them, and it doesn't seem to synergize chemotherapy effects. Is that? I don't know if I'm following your point or not, but I haven't thought about the different temperatures in the lungs significantly. So.
How do you imagine applying heat as a therapeutic in the presence of Paclitaxel? Are you going to send everybody to the hot springs and give them a drug?
So we actually had an interesting project using nanoparticle-based approaches, using iron oxide-based nanoparticles, where we could put, we've done it in animals, you can put animals in magnetic fields and target the nanoparticles to the tumor areas and heat them up. So we've actually done some preliminary work with some collaborators at Hopkins doing that in radiation oncology. So basically, it's only as good as the targeting the nanoparticles, and that's where the tricky part of the business is. The actual particles themselves do indeed heat up, and you can get temperature differentials to work that way. But people have used it, heat is not my idea. I mean, people have used it now in cancer therapies as well. They run, during operations for GI cancers, run warming baths and stuff through the area during surgery. So it's been used in other, and local ones for melanomas. People have used heating boots to heat up the areas during therapy. So there's been a lot of creative ideas. It's really been the thing that's held back the field the most, which is how do you effectively get temperature to raise, and not just raise throughout the whole body, because there's some organs that are actually quite sensitive, normal organs, to temperature differences that don't do very well. So we focused on this nanoparticle approach. I'm very interested in going after some of these modulators like heat shock and cold shock proteins as sort of the molecular switches of temperature. So the idea is, can you get the cell to think it's being heated without actually heating it by modulating some of these proteins like RBM3? By changing its expression, could we get the cell to have the same response to temperature as it would if you heated it? Because the heating part, engineering-wise, is not a trivial concept, but we have been working on that. Other questions?
Well, with the lung, it might be possible.
With the lung, it might be possible. And the other organ that's...
I had a friend who
Yeah.
had lung cancer
Yeah.
and was told that spending time in the sauna
could be helpful, yeah. The other area that's been worked out a lot is the bladder, because the bladder, you can instill fluids into the bladder, and for bladder cancer it can be used. So that was another idea we were going to work on, which is to try to get bladder cancer patients treated that way. But you're right, I mean, that's the original use of saunas and things, was to treat people for a lot of these different conditions. So.
I don't know the traditions about how we do these things. Do we continue with questions here, or let some people stay, or invite people to talk tomorrow mostly?
If there's a talk tomorrow, probably makes sense to just do that.
All right.
All right, thanks, appreciate it.